Welcome back to the second session of the day. We'll now get started with a great panel about the future without animal testing. And again, don't forget to write your questions on the box to your right-hand side for the guided Q&A that we will have by the end of the session. The panel moderation will be performed by my colleague, Dr. Itedal Nanaredwan, CSO at CellLink. So with that said, Itedal, the stage is yours.
Thank you very much, Mariana, and what an honor to be on this panel and have the opportunity to moderate this great and very, very important session. I would like to, first of all, just introduce the panelists, but I will let them tell us about their findings and why they are part of this panel today. So we have Dr. Carol Treasure, we have Dr. Sharon Chandrasekhra, and we have Dr. Siyum Ayuni. So maybe, Carol, maybe you could start by giving a brief introduction about yourself.
Sure. Hi, everyone, and thank you very much for inviting me to take part today in this exciting event. Just to introduce myself briefly, I'm Dr. Carol Treasure. I'm the founder and CEO of Accelerate, which is a completely animal-free testing lab in the UK. But I've been working in the field of replacing animal testing and in vitro toxicology for about 30 years now. And it's been a really, really exciting journey and a real privilege to be involved with such an emerging area that I've always been so passionate about. And I guess ever since I started my career in the pharmaceutical industry, it was always obvious to me that there had to be a better way than animal testing, not just ethically, but scientifically too, building something that was more relevant to humans. And I had an opportunity to do my PhD in the Frame Alternatives Laboratory at Nottingham University here in the UK. And it's interesting now reflecting back on that, because we were using basic 2D cell culture models looking at cytotoxicity, and those models were quite rightly widely criticised for being far too simplistic. And it could be a bit demoralising at the time, thinking, how are we ever going to progress this work? But now looking back on that, I think that that was a really important stepping stone. Because if we hadn't, and not just us, but other groups as well, if we hadn't collectively as a scientific community started off on that work, we wouldn't now have all the explosion of exciting technologies that we're looking at today. Those things eventually led on to tissue engineering, things like bioprinting, the organ on a chip, genomic screening, all the things that we look at now, which are actually very credible replacements and scientifically superior replacements to animal testing. So it's been a really exciting journey. I founded Accelerate in 2008, and we've grown to 20 people here in the UK. And really our mission is to accelerate the world's transition to 100% animal-free testing. And what we really focus on is the implementation of the technologies that are coming out from various groups around the world. It's really important that we take that science and that we're able to provide it to industry, so those methods are actually getting applied taken up and used in regulatory and non-regulatory testing. And just to mention, I have a pet subject is the full replacement of animal-derived components in cell culture models, because we have to ask ourselves what is truly animal-free testing. And a lot of in vitro methods today still use animal-derived components and ultimately require the sacrifice of animals or are using components that are waste products from the meat industry. So that's something that I'm really passionate about. I do believe, just to sum up, that I think the 2020s are really the decade where this is ramping up. The science is really snowballing now into something that's very, very exciting. The pace of change is picking up. So still excited to see where this goes and really happy to be here today.
Thank you very much, Dr. Treasure, and I couldn't agree more. And I'm so happy to have you as part of this panelist. Dr. Ayuni, can you please tell us about yourself?
Okay, I am Siyuma Ayuni, and I'm the Chief Scientific Officer at Matec Corporation, a biotech company. My background is virology, and then I did HIV work. And then I was at Harvard doing my postdoc, and I was involved in molecular biology and cellular immunology work. And during that time, what initiated me to go into this field is that when I was doing HIV research, what happens was I was involved in looking for animal models for HIV, to infect with HIV virus. But there was no animal models. So we tried it for 2-3 years, and still up to now, there is no animal models. And then when Matec was approaching me, one of the interests I had was to bring that concept into, if the animal models are not working, then can I go to the human models and mimic, for example, develop the exocervical tissue model at Matec, and then use that model for HIV infection experiments. So that was one of the motivations that led me to get into the tissue engineering field. And I came to Matec, and one of my first tissue models that I was going to develop was the exocervical tissue model for HIV infection, which I did it with a NIH grant. And with that, we were collaborating with Boston Medical Center, and we were able to infect it with HIV. And that was the closest human model that one can see. So animal models are not that, they can answer some questions, but animal models are not human replacements. We are different from them. And we have a completely different genetic material and so on. So using for toxicological studies and so on, we have to use this in vitro tissue models to answer those questions. And that is one of the things that I did after in 2000, in 2012 was to come up with also a gut model for addressing drug development problems. Some of the companies start with 100,000, 10,000 compounds, and then with the discovery phase, they remain with 250 compounds. And then through the clinical phase, if they are lucky, they will come up with, at the end of the third phase, the clinical phase 3 trial, they come up with one drug. So that is, it takes a long time. And if we have an emergency, there is no time to wait for 12 years to get one drug. So it has to be done immediately. And the proper way of doing it is to come up with some kind of tissue engineering and microphysiological systems to connect different organs together. So one of the things I was involved in doing was to develop the gut model, which we developed it in 2014. And then we collaborate with AstraZeneca to prove the concept that animal models can, using animal models, some of the drugs were passing through. And then most of the 20% of them, were at phase 3 clinical trials, they failed because of toxicity problems. So we were trying to see if these models can be predictive of those drugs. So the first thing we did was we took drugs, we asked them, do you have any drugs that failed in human clinical trial, but passed the animal testing. So they gave us about 4 drugs, and we tested in our models. And then there was also placebos, 7 drugs. And then they gave it to us in a blinded fashion. We did it. And in 100% of the case using these in vitro models, we're capable of predicting those drugs were problematic. So that this type of work was the one which initiated me to get into the field of tissue engineering.
Thank you very much, Dr. Ayuni, and it's very, very inspirational. I think you also gave us hints of what might be the future of microfluidics and so on. Dr. Sharon Chandrasekhra, please introduce yourself.
Hi, thank you so much for the invitation. It's such a pleasure to be here to talk about, obviously, my favorite topic. So I'm Dr. Sharon Chandrasekhra. I'm the founder and Executive Director of the Canadian Centre for Alternatives to Animal Methods and its subsidiary, the Canadian Centre for the Validation of Alternative Methods, located at the University of Windsor in Southern Ontario. So my background is I'm a biochemist and a molecular biologist with specialization in cardiovascular science, and I was a former animal researcher. I've been there, done that, did it all. I worked with a number of animal models of heart failure and diabetes, and throughout that experience in my postdoctoral years, I found out firsthand the colossal limitations of animal models and how, through a couple of personal experiences, when my dad suffered a heart attack, I realized that the work that I was doing was never going to translate to help people like him or all the other patients that I saw in that ward during that time. So I actually walked away from animal research, and I joined a US nonprofit organization, the Physicians Committee for Responsible Medicine. And during that time, I was traveling the world, being exposed to this incredible field. It was a multi-sectoral field with people from academia, industry, government, and nonprofit sectors that work together with a common goal. And so I started thinking that, we're missing something in Canada. So that's what led me to really establish this center. It was merely a thought on the back of a napkin. And so now I have a center with a bold vision to replace animals in Canadian biomedical research, education, and regulatory testing through 21st century science, innovation, and ethics. And so I work with stakeholders from all sectors, nationally and internationally. And I've set up the center with 3 key pillars that I think are quite important for this field. One is a pillar addressing specifically the use of animal-free technologies in biomedical research. So with ours, we're creating 3D tissue models. As you can see, it's my BioX6 here, and that's what I do. All of our tissue models are now 3D bioprinted because 2D cell cultures are not quite cutting it, just like Carol said. And so creating disease in a dish and developing, validating, and promoting the acceptance of these technologies within the basic science community. The other pillar is the regulatory pillar, where I work with Canadian and also international regulators like Health Canada, FDA, EPA, collaborative partners, and other centers for alternatives around the globe to modernize toxicology and chemical safety testing. But my favorite pillar, I must say, is our academic pillar, where we're developing courses and we're creating a graduate program in animal replacement science to train the next generation to think outside the cage. In my personal opinion, the biggest impediment to accepting and replacing animals is a cultural issue. And so if we don't train our next generation to think beyond animal models and to accept all of these amazing technologies at our disposal, then we're doing a disservice. So it is one of my favorite things. We have graduate courses coming up and a number of other training programs. So, I really envision a Canada without animal testing, and hope that we could all work together to move in that direction.
Thank you very much. I couldn't agree more. And I'm envisioning, hopefully, that the world takes the next step to get us all there. Maybe I can start by asking Dr. Treasure a question. Mm-hmm. So originally introduced in the 1950s, the 3 R's principle, replacement, reduction, and refinement, proposed replacing or reducing the use of animals in biomedical research. More than 60 years later, what are the biggest challenges preventing the complete paradigm shift away from the animal testing?
That's a big question to start with. I guess just a few thoughts from my side. There are still some challenges in terms of what we can achieve in vitro with the more complex organ systems. One of the criticisms of in vitro methods has always been that we can't reflect what's happening in the whole animal or the whole human. But I think that there's some really exciting advances there, which we'll probably come on to talk about in terms of linking up the three-dimensional organoids and tissue engineered models to look at how those more complex organ systems interact with each other. That's still a remaining challenge in terms of pushing that research and development through to the implementation phase. How can we actually use those models in the real world applications? I think another one is the long-term survival of the in vitro models, because if we're looking at chronic effects or repeat dose, then that's always a challenge. At the moment, the in vitro models tend to only be relatively short term, so we have to be quick and run the experiments while they're at their peak. So if we can actually keep these models alive in the lab through the kind of time periods that are required for pharmaceutical safety trials, for example, I think that would be a big step forward. So those are some of the technical challenges. I would also say that there's some validation and regulatory challenges, because at the moment, the regulatory pathways globally are very clunky, very old-fashioned. It takes such a long time to get technology validated and approved for use. So I think one of the key challenges is not necessarily a technical one. It's about how can we validate the models and get them through the pipeline and approved by regulators. And I'd say just the final thing is that I think one of the key challenges we face is that in vitro models have always been validated against the animal model. And so we're comparing against a faulty benchmark. You know, we're expected to show that the in vitro model is equivalent to the animal model. And if it isn't, then the in vitro model is considered not good enough. But actually, the animal tests were never validated. There was nothing like this around. You know, now we have much higher expectations. We've raised the bar. And it's good that current models have to be validated thoroughly, but there's a double standard there. And I think it's really important that we just step back from the concept of trying to directly replace an animal test with something that is trying to simulate that. Just do away with that whole concept and start to think about how can we be more creative to directly have a reflection of human physiology. And a lot of the work that's being done with next generation risk assessment, combining in vitro with in silico methods, is really starting to think outside the box. So there's a lot of paradigm shifts that I think are very important.
Thank you very much. Dr. Ayuni, I know Dr. Treasure mentioned the importance of having maybe multiple organs. Is there anything else you would like to add to that and maybe describe some of the work MatTek is doing in that area?
Yes, I will be happy to discuss those points. Some of the points that Carol mentioned, like the longevity of the tissues, the tissue models that we're developing. As MatTek is the leading tissue engineering company globally, and we supply a lot of these tissue models to as far as Europe and Asia. So in terms of longevity, some of our models, we have made a tremendous improvement in increasing the time that this tissue, the shelf life of these tissues, to say for longer time periods, so that people can do it acute and chronic exposure experiments. So like the airway tissue model, the gut tissue model, they can go up to 90 days in culture. So we have done a 42 days, for example, experiment with AstraZeneca. And we were trying to look at not only the drug toxicity, but also drug-drug interaction in that space of time. So those are improvements. Other tissue models which have shorter time span, we are working hard to increase, and probably bringing new technologies like bioprinting and so on may help us to advance the time that we need to keep these tissues in culture. But in terms of microphysiological systems, I think those are key points, because as the DARPA saw it early on in around 2010 and 2012, and they initiated about DARPA and NIH, they initiated about $200 million projects. So we were one of the beneficiaries of those projects at that time. And the whole concept was to put the human on a chip, or that was the whole concept. But at least we have to start from tissue on a chip, and then organ on a chip, and then human on a chip. So those are a small process, but the platforms are there. There are now over 50 companies who can produce these chips that we can grow. They are low throughput, but we can increase, if high throughput formats are coming in, more tissues can be done, and so on. But at this moment, as the high throughput, like us, for example, we produce these tissues in larger quantities, and we were able to use it for validation purposes. Because of that, we have in vitro skin corrosion testing, for example, using our models. The epiderm model is approved by OECD guidelines. And then we have in vitro skin rotation testing, that's also approved. We have in vitro eye rotation testing, that's also approved by OECD. So we have contributed a lot to this, to replace animal models. And for the cosmetic industry, this type of work that we did has changed the whole industry. So now we are heading to the right direction, and EPA is going also by 2035. They are not giving funding for animal experimentation. So they are warning people to line up to alternative methods, and that is encouraging, and they have to pursue that point.
Thank you very much, Dr. Ayuni. Dr. Chandrasekhra, I know that it takes to get the standardized methods, but more importantly, a regulatory approval of those methods. I know you are working a lot within that area, but is there anything we as a community can do to speed that process up?
Sorry, I missed the first part of your, there was a little glitch. I didn't hear your question.
So the time that it gets the standardized methods approved by regulatory instances, is there anything we as a community can do other than the amazing work you are doing? Of course, is there anything else we can do as a community?
Absolutely. So this whole business of replacing animals and moving toward a human biology-based platform, it involves multidisciplinary teams, obviously, to develop the science, and then it also involves multi-sectoral stakeholders. So academics, industry, government, and nonprofit sectors. So we all have to work together because there's a lot to be accomplished. And in terms of, developing new methods and then making sure that they emulate human biology properly, and then going through that pipeline of getting regulatory approval. And I think that those all need to accelerate very quickly, just like Carol and Siam were talking about. And that's where really, in terms of replacing animals in regulatory testing, that's where the bottleneck is, getting all of these technologies that we have at our disposal currently, and getting them approved to a point where you could replace certain tests. And you can see this happening in regulatory risk assessment, right? When you look at the skin sensitization, that used to be measured with like in mice and guinea pigs, the local lymph node assay in mice and the guinea pig maximization test. Those were descriptive whole animal studies that were replaced by something extremely simple, only because we understand the human biology behind it, the mechanisms behind some of these toxicities. And so it's about being creative, replacing that type of in vivo methods with something creative like an in chemical test where you're just looking at protein interactions and a simple 2D human cell-based method. So it is doable. And now the whole community has to come together and play their role. I think the regulators have to step up their game and make the pipeline faster, get this, what type of validation do you want? Let's get it done. And the OECD, they have to move a lot faster. I mean, the skin sensitization defined approach, it took more than 4 years of argumentation around the tables. It could have been done, it should have been done a little bit faster. And now we've learned the lesson. So yes, we need to accelerate that. And the companies, they need to have a vested interest in this field, right? Everyone benefits from it. Drug companies, chemical companies. How long does it take to test, do all the animal tests to get a pesticide approved? So everyone will benefit from working together. And there's no other way to do it because it's not the responsibility of a single sector.
Okay. Thank you very much. Maybe you can continue to answer the next question. So what are the main benefits of ending animal testing?
Oh my goodness. I guess I could just summarize it in one sentence. It's the gift of life. What I see, its benefits, progress in science and advancements in medicine for humankind, and lack of suffering and gift of life for animal kind. It's a win-win situation for everyone. Just think about where we are today. This is the third decade of the 21st century. We're at a point where we have 95% of the drugs that pass animal testing fail in human clinical trials. We do not have safe and effective treatments, disease-modifying treatments for most of the diseases prevalent in our society today. And on the other hand, we have over 85,000 chemicals on the global market, most of which lack full toxicological profiles. If we were to rely on the legacy animal-based methods to evaluate all of these chemicals for all the adverse outcomes that could potentially happen, it will take us several hundred years, cost tens of trillions of dollars, and hundreds of millions of animal lives. This is where we are. It's ridiculous that we, as an advanced species, who carry an iPhone 13 in their back pocket, it's almost like we're going back to, or still using rotary phones when it comes to science and medicine. So everybody across, everybody will benefit from switching to a human biology-based platform.
Thank you very much. Dr. Ayuni, so everyone will benefit. That was the last sentence. I want to ask you, is there one industry that will benefit the most from switching to non-animal models?
I think the cosmetic industry is one that by switching to the alternate methods, the cosmetic industry, which is now basically beneficiary of these alternative methods. And the other one is the pharmaceutical industry, because the pharmaceutical industry, some of the work they are doing in animal models, they are not predictive of human responses, that's period. So they have to come up with additional, alternate methods that can address their questions. For example, if you take the mice and rats, at least they are, for example, 10 times, more tolerant to 37% of drugs in humans. So they are tolerant, and then you bring it to humans, they will be toxic, or the efficacy is not there. And rat and dogs are most probably 4.5 to 100-fold tolerant for concentration of chemotherapeutic drugs. And GI, for example, toxicity is one of the greatest problems for chemotherapeutic drugs. And all these are passing through animal tests, and reducing this drug, attritions by 5%, even by 5% in the phase one clinical development period, they can benefit. It can reduce cost by 5 to 5.7% during the development. That is basically equivalent to almost $100 million that they can save, even if by just changing 5% of the things. In other words, they have to do it in the human. the human, particularly now, primary cells are used like we are using primary cells to make all our tissue models, and they are translatable. They can translate the results they got because they are human cells. The results can be translatable to humans. So they need this type of thing. So the pharmaceutical industry is going to be one of the most benefit. If they change their behavior, if they change their work habit, because I understand most of their data is collected from animals and they don't want to change from there. But the results are not leading them to new innovations either. So they have to pause and think correctly, and then use this advanced in vitro 3D models or organoids or microphysiological systems or bioprinted tissue models or organs to address their questions. With that, I think the pharmaceutical industry will be benefiting from these technologies.
Thank you very much. Dr. Treasure, we're talking about the so where do you think we will be with non-animal models in 10 years, given the scientific developments and technological advancements we've made?
Well, I think the pace of change is certainly accelerating. As I said in my introduction, I think that this decade is really where we're going to make some really amazing advances. I think that certainly the organ on a chip, human on a chip, the development of ever increasing levels of complexity by using bioprinting to help enhance these models is really going to help to drive forward those human relevant systems. I think I'd like to sort of zoom in on one of my pet subjects, if I may, which is about the replacement of animal-derived components. Because at the moment, the vast majority of in vitro testing and non-animal models actually still use animal-derived components. And there's an ethical aspect to that, because the world is very much moving to all things ethical, cruelty-free, vegan. So consumers are actually pulling this from their end and placing a demand on completely animal-free, vegan compliant test systems to match the vegan products that they're buying. There's no point in buying a vegan product if it's been tested using animal components. So that's kind of being pulled through from the consumer end. But I think scientifically, we have to recognize that continuing to use things like fetal bovine serum in vitro models is not only compromising the reproducibility, because each batch of serum is different, and we all as scientists have to do batch testing and constantly make sure that we've got that continuity, but it's compromising the human relevance of the system as well. We know that fetal bovine serum has a very different composition to human serum. Rat liver extracts are being used at the moment to look at metabolism, whereas we really need to shift to not only to human reagents, so using human liver extracts, but actually looking at eradicating those human reagents as well and moving towards a fully defined system. So we absolutely know what we're using in that test. And there are, of course, scientific and ethical drawbacks in using the human reagents as well, but I really feel that the future is very much having a fully chemically defined in vitro system so that we know what's in it. And I think that when we look back in a few decades' time, not only will we be thinking, why did we ever depend on animals for testing, but we'll also look back on the use of animal-derived components in those in vitro tests as a fairly primitive first step. And I would just like to appeal to everybody who's here listening today that if you are developing methods from the beginning, please, please invest the time and effort to make them chemically defined from the outset and eradicate all animal-derived components. Because we at Accelerate, we've had to go back and adapt so many methods to animal-free conditions after the event, after they've been accepted by OECD, to make them suitable for our animal product-free lab to help our clients. And I think that if we're really taking a long view, we know that these methods can take years and years to get through the regulatory system, which Charu, you alluded to that, and that's very important that they cut that down. But knowing that those tests take years to be approved, we really need to be looking ahead into the future. And from the outset, let's future-proof our new methods. Let's future-proof any assays that are being developed by going animal-free from the beginning.
Thank you very much, Dr. Treasure. Dr. Sharon Chandrasekhra, I have a question for you. We've been talking very high level, but I know many of the scientists in the audience are curious about how we can make improvements to the non-animal models if we should tackle the problems of making more complex living organisms. And I think what we are curious about, what are the mechanisms, what are the physiological important features that we need to mimic in our in vitro models?
So when it comes to, well, there are a number of different aspects. And I want to start off by saying that when people are thinking about replacing animal models with human biology-based methods, it's not, I mean, there's no single end-all, be-all, all-encompassing magical technology that could replace it all, right? I find that every time I talk to scientists who are trying to make that switch and giving up because they're like, oh, it doesn't work for my system. So it's really important to understand that we need to recapitulate human biology at every level of biological complexity, depending on what question you're asking in science. So if you're looking at genomics, you don't need to worry about, population level studies on your first. So if you're looking at like single cell RNA sequencing, for example, then you're not so concerned about other factors in your system. So you could look at everything that happens at the molecular level, cellular level, organ level, tissue level, organism and population level, and integrate that data as you see fit for the purpose of your hypothesis or your experimentation. So in terms of recreating that biological complexity, you're going to need to use different types of in vitro models. So if you want the complexity, the kind of thing that I'm really interested in, moving away from that two-dimensional cell culture, cell culturing that we've had for the past 100 years and moving it into a 3D system. And so what we've discovered, we have a number of, we have 5 different models that we're working on currently. So 3D bioprinted human liver, tissue, lung, kidney, GI, and we're also working on a blood-brain barrier model. The difference that we see, just taking simple cell lines like HepG2, that has been in existence forever and that's been used extensively, we take these two-dimensional, 2D grown flat on a petri dish cells and put them through the BioX6. Everything changes. The physiology changes, the expression of the drug metabolizing enzymes, the function, albumin secretion, everything absolutely changes. They are far more physiologically relevant. They mimic human biology better. So we need to be creative, and then, what do you use that for? And then at the same time, could you then work with a surgeon in the hospital nearby to obtain human primary tissue samples from different stages of development, from, a small child to a senescent stage, and under different disease conditions, under different stages. So it's about harnessing all of the different components that we have access to, whether they are fresh tissues, whether they're engineered tissues, and whether it's epidemiological data or biomonitoring data that we can incorporate and get an understanding of that human biological complexity and combining them in creative ways. So it's not a one-to-one replacement for anything. And a lot of people think that it's a one-to-one replacement, but it isn't. What are you interested in? What biological thing do you want to look at? And then how do I gather the techniques that I need and the biomaterials that I need, whether it's human saliva or blood or a tissue sample, and how do I incorporate that creatively into my model?
There is a question from the audience that is very related that I would like to ask since we're talking about it. So what do you think about dynamic culture devices that allow you to stretch and compress your cells?
I think dynamic cultures are the next game changer. It's absolutely essential. So static cultures are limited, and especially, they're limited in terms of the kind of physiological properties that get exerted on your tissue models. So I think the next big thing is to really move away from static culture into dynamic micro or millifluidic cultures to give the tissues, especially if it's, if you're looking at the lung, you do want to mimic the contractions or even the peristaltic movement of the gut. It's really important to mimic those, and to even in regular cell culture, if you have a 3D model, to just have continuous fluid flow, it absolutely makes it more physiologically relevant. And if you could incorporate then on top of it, right, the next big thing is to incorporate immune cells, to incorporate nerve innervation into your tissue models and circulation, and to have biologically relevant media and tissue and formulations, animal-free, just like Carol said, obviously, to mimic systemic physiology.
Thank you. Dr. Ayuni, this is a question from the audience as well. Have we resolved the scalability and the standardization of in vitro models to eliminate animal models? And if not, what are the remaining challenges?
Yeah, I think the remaining, scalability is one of the problem in this area, in the field. So basically you have to know, to have that scalability issues to resolve it, you have to know, say, for example, if we are doing constructing epidermis, for example, with immune cells, you cannot come up and load the immune cells into this type of tissue models. Say, for example, in the skin, you have only 2 to 4% of Langerhans cells in there as immune cell component to grab whatever antigens are coming in or pathogens are coming in. So you have to know exactly that, and then you have to do it proportionally. You have to work it proportional, but some people are working 1 to 1,000 in this one, but the surface area probably you can come up with the scalabilities. What we're trying to do is we look for the surface area of the tissue, and then we are bringing down to the surface area of the tissue models that we are going to make. And from there, we can have some kind of proportionality to start with. But in the meantime, the composition of the cells and so on and so on, you have to have a good biological knowledge to put it in the right proportion. If it's not in the right proportion, the models are going to be skewed also. So we're aware about those things. But scalability is a big problem. And in the meantime, when we are doing, particularly when we are making these microphysiological systems, there is a big movement to come up with a universal type of media. Now in our body, it's blood, but in tissue culture systems, you have hundreds of media, different media that you use. For a kidney, you use one media. For liver, you'll use one media. For skin, you'll use one media. But in the body, you don't have that. You have only one. So there is a lot movement to go to a universal type of media formulation. But even if we don't reach fully, at least if we can encompass the most important organs in one media, if they can survive for certain time periods, that will be the greatest achievement to science. So there is a movement in that area. When those things are improved, the scalability will be improved, and the tissue formation will be improved. And as Charu said, you need the right, matrix protein. You need the right, substrate to culture these cells. If you don't have the right substrate, then they will change into something else. So that is critical. So those are type of things you provide that, like the the bioprinting, the ink that BICO is providing. Those will give us the right substrate to the right organ formation, and then you have improved it, and that will answer questions to the scientific community. But the scalability issue is probably it will take probably 5 to 10 years to come up with a defined model to use it.
Thank you very much, Dr. Ayuni. So I know we are getting to the end of our session, but I really need to end with this excellent question from the audience. And I'll Dr. Treasure, if you don't mind. So the question is there any recommendations of how researchers in developing countries can also benefit from advanced tissue models despite the challenges of higher cost compared to 2D cell culture?
Well, I think that we have a global responsibility as the whole scientific community to make sure that the whole of society in every country can move forward together with this. And I think that there are a number of ways to do that. I think just taking it down to a scientific level in the lab, I know that sometimes it can really make sense to do some initial proof of concept work using the lower cost two-dimensional cultures. So depending on the scientific question, of course, sometimes that will help, sometimes it won't. But then to do that as a preliminary step and then work up to the 3D cultures later on in the project can be really beneficial. In terms of providing the more complex models internationally, I think it's critical that we do look at this together as a community, because it has an impact not only on the evolution of science in different geographical areas, but in the regulatory acceptance of non-animal methods. If we're to persuade the regulators in all of those countries to change the law to ban animal testing for different sectors and to approve the use of the non-animal methods, there has to be a local ability to actually apply those methods. So I think that the governments and industry in all of those countries have an interest, if they want to keep up with the global trend towards non-animal testing, to actually keep up with developing those technologies in their countries. So I think that if we can work with governments, industry, regulators to try and get more funding into those areas, we need those countries to be able to practically apply the models if we're going to change the regulations. And maybe it's about collaborating with a partner overseas, or collaborating with a supplier. I think it would be great to have a whole separate discussion on this topic, to have a brainstorm on how can we pull together to share the resources that we have and make sure that this is a truly global effort. It's a tricky one to answer, but those are my thoughts.
No, I couldn't agree more. It is a very tricky question, but it's also our responsibility in the industry and also as a scientific community to make sure that the advanced and accurate models become available, hopefully everywhere and approved everywhere. I want to express my sincere gratitude to all of you for being part of this panel. I really, really enjoyed talking to you, and I'm really looking forward to continuing, of course, this discussion. We will have to do it offline, and hopefully next time we meet. Thank you very much for your time, and thank you for joining. And at this point, I want to thank the audience for tuning in. And with that, I would like to hand over to my colleague, Mariana.
Thank you.
Thank you, Vitadal. And also thank you for all the invited speakers. And of course, this was a very good discussion. So thank you very much for joining us today. And we'll now give everyone that is attending the partnership conference today the ability to network and meet like-minded people attending the conference. So the speed networking session will now get started, and you will find the session on the menu on the left of your screen. So thank you very much for attending this session, and I'll see you there.
All right. Hello, everyone. My name is Gardner Butterworth. I'm the head of sales for Selling Bioprinting in the Americas, Middle East, and for our daughter company, Advanced Biomatrix. Today, I have the absolute pleasure of introducing our next speaking panel, Advances in Tissue Engineering. Our panel moderator for the session is Reggie St. Louis, a tissue engineer here at Selling. Just a quick note to the audience that you can submit questions to the panelists at any time throughout the session by typing in the Q&A panel to the right of your screen, and we'll take some time at the end of the session to answer your questions. With that, I will now turn it over to you, Reggie. Have at it.
All right, wonderful. Good morning, everyone. It is so great to have you here in this session today. As Gardner introduced, my name is Reggie St. Louis. Here at Selling, I work a lot with our bioprinting services and trying to, offer our technologies and help people get onboarded and started up in the field of bioprinting. So with us today, we have a phenomenal panel that actually spans quite a good breadth of the different applications that we actually have in the fields of tissue engineering and bioprinting more specifically. So the way I'd like to start this off is if we could just go around and